Hello my dear students, welcome to 12th virtual class of the topic Molecular Basis of Inheritance. Today we are going to learn a very important and interesting topic called DNA Fingerprinting. This is the last topic under this chapter. This topic is having a lots of applications. DNA fingerprinting is one of the technique which is used to identify the nucleotide sequence of DNA. Remember, the nucleotide sequence of the DNA is unique to each individual. No two individuals are having similar type of pattern of nucleotide sequence, which is unique to each personality. Just like we have unique fingerprints, unique retina. Okay. So similarly, the pattern of nucleotide sequence is also unique to each personality. That's why it is commonly called DNA profiling. The profile which is unique to each personality. Similarly, this technique is also called DNA profiling because the pattern of nucleotide sequence is unique. And this technique was developed in the year 1984 by one scientist by name Jeffrey, who is British genetist, who devised this technique by using the satellite DNA as probe. The satellite DNA is used as a probe, and he designed this technique of identifying the pattern of nucleotide sequence present in each individual's DNA. So it is Jeffrey who used the mini satellites or the DNA probe in the form of a variable number of tandem repeats. He used the probe in the form of a variable number of tandem repeats, which is commonly called VNTRs or mini satellite. So here the DNA probe or satellite DNA, which is used as a probe for identifying the unique pattern of nucleotide sequence present in a human DNA. Remember, in the previous topic called Human Genome Project, we have come across there are nearly 100 to 1000 repetitive DNA sequences present in a human DNA. So this repetitive DNA which is found scattered throughout the DNA, and that will found ultimately scattered in chromosomes also. So this repetitive DNA which will found repeated several times, thousands of times it is repeated and it is distributed all over the DNA. The pattern of distribution of this repetitive DNA, the number and the position which is unique to each personality. No two individuals show the same type of repetitive DNA or the DNA what we commonly link with VNTRs, that will not show the similarity between the two individuals. No two individuals show similar type of this repetitive DNA in the form of number or as well as in the form of position scattered in the DNA as well as in a chromosome. So by taking this advantage, Jeffrey designed this technique where identifying this repetitive DNA sequence in the DNA as well as in the chromosomes which is inheriting from the parents to their offspring. As we know, chromosomes will inherit from parents to their offspring, while it is inheriting the characters present in the paternal chromosome as well as maternal chromosome that also inherited or passes to their offspring. So similarly, these repetitive DNA which are found in each individual in a specific number and in a specific position, some of these repetitive DNA or the part of that repetitive DNA will be passing from the parents to their offspring. By taking this advantage, the DNA profiling or DNA fingerprinting will be done, where DNA matching, or commonly we heard about DNA testing. It is nothing but the DNA fingerprinting or DNA profiling, in which the DNA from one person is collected and that will be tries to match us with other suspected person DNA. As I mentioned in the beginning, it is having a vast applications. So we'll see the applications at the end. So here the repetitive DNA will be linked or tagged with these DNA probes, or what are commonly called the VLTRs, the satellite DNA or mini satellite DNA, that is acting as a probe that will be tagged or linked with these repetitive DNA, which is found scattered throughout the DNA, and that is passing to the chromosome, and that will be helping out to know the locations and the number of these repetitive segments or repetitive DNA which is present in a human DNA. We'll see what are the steps which are involved here for DNA fingerprinting. Steps for DNA fingerprinting. As we discussed, what is DNA fingerprinting? As the repetitive DNA sequence, which are unique to each personality, which is inheriting from the parents to their offspring. So these repetitive DNA sequence are what we call as a mini satellite or variable number of tandem repeats, which show high degree of polymorphism, which is going to be inherited from the parents to the offspring. So by taking this advantage, there is technique has been developed. To carry on this technique, there are a number of steps involved. So first and foremost, the collection of sample of DNA. So usually this particular technique is widely used for the detection of the crimes. So crime will be solved, or various criminals will be captured by using this technique, DNA fingerprinting. So for that, the sample will be collected from the vicinity of the crime. The sample may be hair or any body tissues, or it may be any body fluids or the various secretions from the body. Any sample which is collected from the vicinity of crime, as acting as a source for the DNA from this sample. So the sample may be hair, or it may be blood also, where the WBCs can be used, or it may be the tissues, etc. can be extracted or collected from the vicinity of the crime. Then from this sample, isolation of DNA. The DNA will be going to isolate it from the tissue or from the sample which has been collected from the vicinity of crime or from any other place. So from the sample, the DNA will be collected by using density grade centrifugation, by treating with chilled ethanol, where there are a number of techniques for that. So by using the specific technique, the DNA will be isolated from that sample. Followed to that, the fragments will be made. Fragments of DNA will be made. To get the fragments of DNA will be treated with certain restriction enzymes, which cuts the DNA into number of fragments. So the fragments of DNA is very, very important. Without fragmenting, we cannot identify these DNTRs or the mini satellites. Then followed to this fragments. Then the third step, separation of DNA fragments by using a technique called gel electrophoresis. There is a technique called gel electrophoresis technique. By using this technique, the DNA fragments will be separated in the presence of electrolyte. As DNA is negatively charged molecule, this negatively charged molecule will be separated in the presence of electricity and it will run towards the anode. Let me show one small diagram here. The slab called agarose gel slab will be prepared. In this agarose gel slab, DNA will be loaded. The DNA fragments will be loaded at one end, and this entire slab will be placed in an electrophoretic tank, what we call gel electrophoretic tank, in which there is a presence of electrolyte and it is connected to anode and cathode. As it will be run in the presence of electricity, that is, the DNA fragments will move towards anode. Based on the number of this VLP or the mini satellites, the biggest fragment or the largest fragment will be found in the early stage. The smallest fragment will be found at the farthest end. It will be moved by capillary action in an agarose gel. So separation of these fragments will be done by using a technique called gel electrophoresis. So follow to this separation, transfer of DNA fragments to nitrocellulose sheet or nylon sheet by a technique called Southern blotting technique. Southern blotting technique will be used. By using Southern blotting technique, the DNA fragments will be transferred to nitrocellulose sheet or also nylon sheet can be used. So follow to this transfer, these fragments will be treated with DNA probe. The DNA probe is specific in nature, that is going to link to the particular repetitive DNA sequence present in the DNA fragments that will be treated to these fragments. Follow to that transfer, the nitrocellulose sheet which receives this fragment. So follow to this transfer. In the next step, the DNA fragments will be treated with DNA probe. Follow to this treatment, it will be observed under X-ray chamber. Observing under X-ray chamber where autoradiograph will be developed, bearing all the number of bands of the DNA which is present in the form of fragments, which possessing the number of repetitive sequence or what we call the mini satellite, then the position of this mini satellite and the number which is found in particular location, all the detail, the pattern of sequence of that nucleotides present in the form of repetitive DNA sequence, that will be matched, that repetitive DNA sequence will be matched, or it will be going through autoradiograph. I'll repeat. Once the DNA fragments will be treated with DNA probe, it will be autoradiographed under X-ray chamber, where it will be observed the number of VNTRs which has been, or mini satellites which has been found at particular location, and the number of that all details will be collected in the form of an autoradiograph. This autoradiograph is called as an DNA profile, which show the pattern of nucleotide sequence present in the form of a repetitive DNA in that particular sample. In other words, autoradiograph is a profile, DNA profile of that sample. Okay. Now this autoradiograph will be matched with suspected person's DNA. The suspected person DNA will be collected, and that also will be done in the same steps, and that autoradiograph also will be developed. Now these two autoradiographies will be matched, where the number of VNTR, the mini satellite, the position, the number, everything will be matches in all the 46 chromosomes. If all the 46 chromosomes, their VNTRs and the mini satellites will be matches, then it will be declared that the sample DNA and the suspected person DNA is one and the same. Then that person might be involved in a crime, and that person can be catched. So it is having a vast applications. Now if you see the applications of DNA fingerprinting, this technique is having a vast application. The number of applications. This is also asking for 5 marks. One of the major question. The other one is explain the steps involved in DNA fingerprinting. That is what I have explained here. So many steps you have to explain. Similarly, the applications of DNA fingerprinting. This is another one, a major question which is asking in the board examination for 5 marks or 3 marks sometimes. So as I mentioned, there are a number of applications. This technique called DNA fingerprinting is one of the powerful tool using in the field of forensic sciences to catch hold the criminals, to solve the various crimes like murders, rapes, etc. There are a number of crimes will be solved by using this particular technique, what we call DNA fingerprinting. Similarly, to reunite the lost children to their respective parents, this technique is playing a very important role. As I mentioned in the previous, there are a number of repetitive DNA sequence which is inheriting from the parents to their offspring, which show high degree of polymorphism. This is playing a role to reunite the lost children to their respective parents by matching these mini satellites, or what we call the repetitive DNA, which has been inherited from the parents to their offsprings. Similarly, to claim the child by the parents, like in other words, to solve various maternity and paternity issues. Most of the places, one child will be claimed by more than one couple. In that case, DNA testing will be done from both the parent, both the couple and the child. In that case, exact, the VNTRs are what called as the DNA profile will be matched with the persons who is claiming with the child. So if it matches, it gives an 100% identification. So that is another one of the major application where the paternity and maternity issues will be going to solve. Similarly, the DNA fingerprinting is used to claim the dead bodies, to identify the dead bodies, sometimes due to natural calamities or due to some accidental deaths, or it may be volcanic eruption, or it may be any train accidents or plane crashes, etc. Sometimes the bodies are not in a condition to identify. So in that cases, the DNA profiling or DNA testing will be done. If somebody is claiming for that particular body, their DNA will be collected, and the dead body's DNA will be collected and tries to match us with these VNTRs. and if it matches, then it can be claimed that that particular body is belong to that particular family. So the dead bodies can be identified. Then immigrant disputes, which is also one of the major issue in most of the countries, especially in the border areas of the country. There are a number of immigrants. That dispute also can be solved by using the DNA fingerprinting. Similarly, this DNA fingerprinting is also playing an important role in the population to identify the population diversity, to check the population diversity. The frequencies of this DNA polymorphism is very high. That is playing a very important role to identify or to determine the genetic population or population diversity. So like that, if you go on discussing the applications of DNA fingerprinting, it is very vast list. So some of these are the major applications of DNA fingerprinting, which is playing a very important role to solve various issues of the man in the form of social issues, which is going to solve by using DNA fingerprinting. So with this, we have come to an end of this chapter, what we called molecular basis of inheritance. This chapter is totally dealing with genetic material, what we call DNA or RNA, and number of activities related to that, how the metabolism will be regulated by DNA. As we know, DNA is genetic material which is controlling almost all the characters in an organism, how it will control and how it will be going to done all the activities or metabolic activities of the cell that we have seen in this chapter. So it is one of the most important chapter dealing with the metabolism and genetic contents. There is DNA and RNA and their components, etc. The detailed aspects we have discussed. So in the next classes, we'll come out with few more new topics and we'll try to understand. So with this, I'll conclude here. This topic is come to an end. So thank you very much. Be safe. Have a good day.